What are those chemicals? What are those chemicals? ఏ కెమికల్స్ యూజ్ చేయొచ్చు? These are the devices where we will use chemicals, and we cannot use a simple chemical for purifying the enzyme. We need many things. We need many things. So the main objective is purification. So purification, generally enzyme is purified from its contaminating proteins from the mixture, because after the cell lysis you will get all the things that are present in the inside the cell. All the things that are present inside the cell, it contains proteins, carbohydrates, cell organelles, all those things. So we have to follow step by step process. So first of all, after cell lysis we will do a small centrifugation wherein we will remove the unlysed or the cell debris. All these are removed, and after that you will collect the supernatant. You will collect the supernatant which is containing all these nucleic acids and proteins and carbohydrates, all those things. So after that we will prepare. So for that first we need enzyme preparation that have been clarified and concentrated. Clarified means all these things, the contaminated cell debris, all those things, they have to be released and they have to be concentrated, and they are now in suitable state for further purification. So for purification we apply all these methods, either the chromatographic method, majorly chromatographic method we use. The best example are ion exchange chromatography, affinity or gel filtration or size exclusion. Before going into that, actually we will do one more thing, that is, have you ever heard about the ammonium sulfate precipitation? Ammonium sulfate precipitation. Generally people will do initial step as ammonium sulfate precipitation, where it precipitates out the protein. Precipitates out the protein, and ammonium sulfate precipitation will be done in different stages, like by using 20%, by using 40%, by using 60%, by using 80% ammonium sulfate. So depending upon the size of the proteins or the solubility of these proteins with ammonium sulfate, some of the proteins they will get precipitated in 20%. When you use 20% ammonium sulfate, some of the proteins will get precipitated, and some of the proteins will get precipitated at 40%, and some at 60%, and some 80%. Most of the 80% proteins, they will contain the bigger proteins. That means they require more amount of ammonium sulfate for their solubilization. You might have heard about the salting in and salting out. Salting in, salting out. You know that? Salting in and salting out. So whenever you are using this ammonium, any salts. So salting in is which is responsible for the solubilization of the protein. Salting out means precipitating the protein, wherein you will use high amount of salt. So under low amount of salt, this salt interacts with the protein. Because ammonium sulfate. So ammonium is a positive charge, sulfate is a negative charge. So these charged molecules, they will interact with the charged amino acids of the protein. Solubilization is nothing but interacting with this salt. And whenever you use high amount of salt, what happens? These salt molecules, instead of interacting with the protein, they will interact with the water. That means all these proteins, they will get precipitated. So that we call it as salting out. Whenever you are using low amount of salt, so which interacts with the protein, we call it as salting in. So by this, the protein gets solubilized. So there is the first step wherein people will use during the purification protocol. So our main aim is to purify the enzyme. So to purify the enzyme means we have to get only our protein and we have to delete all the things. We have to remove all the things. So while doing your plasmid isolation, what you will do? So you want to isolate only the DNA, plasmid DNA. You have to remove all the remaining contaminants. It may be a chromosomal DNA, or it may be RNA, it may be a protein, it may be carbohydrate, it may be a lipid. So for that you will do step by step. In the initial step, you will do centrifugation wherein the cell debris is removed. Cell debris, that means after lysis you will get the membrane, the lysine membrane. That membrane we call it as the cell debris. So that is removed. After that you will get all the components, and during that period you will use several things like treatment with chloroform, isoamyl alcohol. So during that you will get 3 interphases. So down one is the phenol layer, and in between the middle one is the chloroform, wherein you will see all the proteins get precipitated at interface. In the aqueous phase you will see your DNA. So that means the separation is right there. In the similar manner, we will go by step, wherein while doing the ammonium sulfate fractionation, you can remove all the things and only will get your protein get solubilized in specific percentage, either it may be 20, 40, 68. So after doing ammonium sulfate fractionation, then what you have to do? You have to check your enzyme is present or not. Suppose you assume that you are isolating or you are purifying an enzyme called trypsin. Trypsin enzyme again isolate cheskunnam. So what is the action of the trypsin? Trypsin is a protease. Trypsin is a protease. While telling about the commercial functions of enzymes, I told that trypsin is used to clear the cloud or the cataract that is formed inside the eye. Because it is a protein in nature, this trypsin enzyme is a protease which hydrolyzes the protein. So the function of the trypsin is protease. So it hydrolyzes the protein. So after cell resorption, you will get all the components from the cell, and then first step you have to check is whether your enzyme is there or not. So for that we will do enzymatic activity. As it is a protease, we will go and check the specific substrate for the trypsin, so that we come to know that trypsin is present and it is active. Then you will go for ammonium sulfate fractionation, and during this ammonium sulfate fractionation I told you can do several fractions, 20%, 40%, 60. And in all these fractions we check for activity. We check for this enzyme activity with the same substrate. Then you find which fraction is having more. You go for that percentage. It may be 20, 40, 60. Whatever it may be. So you go to that percentage which shows higher activity, and you take out that percentage, and that fraction you take for further purification process. Because that fraction is containing more amount of your desired enzyme. It contains more amount of your desired enzyme, so that whenever you go for purification with that fraction, you will be having a chances more to purify your desired enzyme. So the first of all people will use ion exchange chromatography. Ion exchange chromatography. It is also a simple technique where the charge plays an important role. Because protein is made up of all amino acids, and many of these amino acids are charged. So automatically protein is also having charge. Okay. So in this ion exchange chromatography, you know that you will be having a resin-like structure which is producing charges on their surface. They produce charges on their surface. So whenever you pass these protein molecules through this, so these proteins, they will interact with the charges of this resin. by ionic interactions. And after that we will do elution. And the protein which is binding more to the resin, it elutes last. And the protein which binds less will elute first. So during the purification of this step, so the first and last. So whether the hydrophobic molecule will elute first or the hydrophilic molecule will elute first. Hydrophobic protein will elute first or the hydrophilic protein will elute first. So do you know the principle? So I will change chromatin. We have studied. So I am also telling you. So the proteins, they are having charge. So they will interact with the resins. So I said that more charge containing protein will bind more with the resin, and the less charged molecule will bind less. So the less charge will elute first and more charge will elute last. So this is the principle. So based on this principle, now you say that hydrophilic protein will elute first or hydrophobic protein will elute first. Hydrophobic protein will elute. In terms of amino acid structure. So hydrophobic protein means which contains more amount of hydrophobic amino acids. Hydrophilic means which contains more amount of hydrophilic amino acids or charged amino acids. Now you say which one elutes first and which one elutes last. So hydrophobic elutes first because they are having more amount of hydrophobic amino acids, and these hydrophobic amino acids, they don't have ionic charges. So their interaction with the resin is very less. So they elute first. And the hydrophilic proteins means which contains more amount of hydrophilic amino acids, that means charged amino acids. So they interact more with resin, and so they elute last. So that is the principle. And in this we have cation exchange chromatography or anion exchange chromatography. So cation is, what is the charge of the cation? Positively charged is the cation. So if you want to do cation exchange resin, cation exchange chromatography, what type of resin would have to use? The resin should be anionic. That means it should possess negative charge on the surface. So whenever it possesses negative charge on the surface, these positively charged proteins, they will interact. And we are saying cation exchange. On the resin, we are having anions, negatively charged, and on the surface of this, the cations are exchanged. So now you pour all the fraction, that is, suppose you assume that you are having more activity of trypsin in 40% ammonium sulfate fraction. You take the fraction and you load onto the column containing this anion resin and allow the protein to flow through this. So whatever the positively charged proteins are there, so they will interact according to their charge density or according to their number of ionic interactions. After that, how to elute? So then we have to use a buffer containing more amount of positive charged ions. Positively charged ions. Then these ions, they will come and they displace the proteins that are attached to the resin, because The buffer which we are adding contains more amount of positive charge, or more or high number of positive charges. They will automatically displace the proteins that are bound to the resin, so that they are eluted. They are eluted. You understand? So you take this. You assume that this is the resin containing all negative charges. And you see the protein that is having positive charge will get bound. Will bind this protein. So this is the column assume that this is the column. Now the process is over, and now we want to elute these things. You want to elute these things. So you want to elute protein. So for this we will use a buffer containing more number of positive charges. More number of positive charges. Simply we call it as high ionic strength. High ionic strength. Whenever we use that high ionic strength, then so in the first, suppose you assume that you have added 1 millimolar sodium chloride solution, 1 millimolar sodium chloride solution, or 1 millimolar imidazole solution. Like that, we will use some ionic strength buffers. So when we have used 1 millimolar, so the buffer passes through this, and this 1 millimolar would be able to replace this one, so that this protein is eluted first. This protein is eluted first. That means with this ionic strength, only the less bound proteins are eluted. And in order to remove this, we need what we need. Because of this 1 millimolar strength, we could be able to elute only this protein, and it is very least bound when compared with these two. So if you want to elute these two, so what we have to do? Good. So we have to increase. We have to increase the strength of the ions. We have to increase the strength of the ions. At this 5 millimolar, I could be able to release this also. I could be able to release this also, but not this. So if you want to release this, still more than this. So you have to use like this. Then you will get complete. So when we have used this, we got some fractions. We have collected some fractions, and then go over activity. Check for activity. So activity is there. No. And then you have eluted with this, so that this has come out. So again you check that fraction. Check for activity. Check for activity. And then you do this. Again you check for activity. That means you collect all the fractions. Collect all the fractions and check for activity. And check for activity. And you make a mark which fraction is having activity. Is having activity. And you record them. You record all these things in your book so that it becomes SOP. Standardized Operating Protocol. Standard Operating Protocol. That means whenever you are doing again the same enzyme purification, it is very simple. You can directly go. Suppose you assume that if it is coming in 5 millimolar, instead of going with 10 millimolar or 1 millimolar, you can directly go to the 5 millimolar. You collect the fractions, and all these fractions are contained in these ions. So that is standardization. Initially, if you are doing, then you have to collect all the fractions. fraction, and we have to determine which fraction is having activity. Once you determine that, yeah, it's over. So then take those fractions. Take those fractions. I have taken only just 3 proteins as an example. But if you see in the cell lysate, you will be having hundreds of proteins. Hundreds of proteins or hundreds of enzymes. So there is a possibility that in your fraction, along with your enzyme, so many enzymes or so many proteins are also present. So in that fraction which you are having activity, then you collect that fraction. Again, we can go for anion exchange chromatography. Anion exchange chromatography. So anion exchange chromatography is also very simple, where you will see the resin is having all positive charges, all positive charges, and the protein should be in negative charge. How you will bring the protein to negative charge? So you have to select a buffer. You have to select a buffer wherein your protein will become negatively charged. Negatively charged at a particular pH. At a particular pH. So you know that isoelectric point. Isoelectric point. What is the isoelectric point? Isoelectric pH or isoelectric point. What is deuteride? Deuterides contain isoelectric point. So what is deuteride? Which contains both positive and negative. So what is the net charge on the net charge? So isoelectric pH or isoelectric point is a particular pH at which the net charge on the protein is 0. Deuteride means which contains equal number of positive and equal number of negative charges. So then the net charge will become 0. In electrophoresis, if you want to move a biomolecule, then it should be charged. You will be doing across the electrophoresis. You are running the DNA. So DNA moves from which pole to which pole? Negative to positive. Why? Negatively charged. So in the presence of electrophoresis or electrical field, so this moves from negative pole to positive pole because it contains all negative charges. So it moves towards the positive pole. In the similar manner, if you do electrophoresis of a protein, it contains more number of charges. It contains mixed. It contains both positive charge and negative charge. Both positive and negative charges. And during isoelectrofocusing, you might be heard about this. Isoelectrofocusing, wherein these proteins, they will move and they will stop at a particular position, and that position we call it as isoelectric pH. Isoelectric pH, where the net charge is 0. So like that, these are enzymes or proteins, they are having charges. And depending upon the buffer activity, the charges of the protein is changed. That's why we can use both anion exchange chromatography and the cation exchange chromatography. Based upon the buffer composition, you can change the charges on that. Change the charges on that so that we can apply both techniques for the purification of the enzyme. You can see cation exchange chromatography, wherein you will see the resin is having negatively charged anions and the positively charged cations, they can be exchanged on the surface of this. So here you can see a protein which is having very least binding and medium binding and a high binding. So they will least binding, they are eluted first compared to these things, and like this you will be eluted. Because you can see here they are using 400 millimolar of NaCl and here 700 millimolar of NaCl, and here they are using still more. Still more. That means ionic strength is increased in order to release. In order to release the bound proteins. Bound proteins. And anion exchange chromatography. So in this, anions are exchanged on a cation resin. So here the resin, this is the resin which contains more number of cations, and whenever the proteins are loaded onto this, the cation proteins, the anion proteins, they bound to this cations. They bound to this cations. Here one is there, one is there. And the positively charged proteins, they are eluted out. They don't bind. They don't bind because these are like pulses, so they repel each other, and so they are won't bind. And these bound proteins later simply by means of increasing the strength, they can be released now. They can be released. So this is about this anion exchange chromatography. And next we have the affinity chromatography. Affinity chromatography. Affinity. Here you will be seeing, because you know that enzymes, they bind to they are specific in nature. They bind only to their substrate. Only to their substrate, otherwise they won't bind. Because enzyme substrate interaction is a specific interaction. So one enzyme won't go and bind to the other enzyme substrate. So it will bind to only its substrate. So here people, what they did is, they have prepared a resin. They prepared a resin and which has this substrate on the surface. Just like we have shown the charges, this resin is bound with the substrate. Bound with the substrate. So whenever you are producing substrate for a particular enzyme, then you pass your protein solution, you pass your protein solution through the column, and only the specific enzyme to that substrate will bind, remaining all other eluted out. Remaining all other eluted out. Because this happens only due to the specificity of the enzyme substrate complex. Enzyme substrate complex, or sometimes even people can use enzyme ligand complex. Enzyme ligand complex. So in your molecular biology, you have studied about the expression of the proteins using histidine tag, histag proteins ani manaku chadivaru. If you could able to remember, in your molecular biology in the last 4th unit, you have studied expression of the proteins using tags, wherein you have studied about the histidine tag. That means whenever you are expressing your protein, you will be adding histidine tag. Histidine tag. That histidine tag is already present in the vector, so that whenever you express your protein along with the histidine tag, it is expressed. And these histag proteins can be purified. By using affinity chromatography. Example I am telling you. By using affinity chromatography. In the affinity chromatography, they will prepare a resin and it is coated with nickel. The metal nickel ions. And to these nickel ions, only the histidine proteins will get bind. Only the histidine proteins which contain more amount of histidine, they will bind and remaining all other are eluted out. And now we have expressed our protein with the histidine tag. So if you are having the histidine tag, only the histidine tag proteins they will bind to this nickel and remaining are all eluted. And after that, by using the high ion extent, you can purify your protein. You can purify your protein. That is the importance of the affinity chromatography. Affinity chromatography. In order to purify your desired enzyme, either you can use its particular substrate, or if you are having a tag like histidine, you can use nickel. You can use nickel. And one more thing is, we have immunochromatography. Just like affinity chromatography, immuno, wherein people will use the resin which is coated with antibody. Which is coated with antibody. And you all know that antigen, antibody, they will interact with each other. So whenever the antibody is coated and it will pass the solution, the specific antigen is nothing but a protein. It may be your enzyme also. It may be an enzyme also. So if you use that specific antibody, so the corresponding antigen will bind to that and that can be purified. That can be purified. So this is the basic principle and the methodology for this affinity chromatography. So you will pass the solution, mixture of proteins, because this is a resin, and you can see that unwanted proteins are washed off through the towel and only the desired. So this is the desired. So the desired proteins will bind, and then you will add the solution of ligand again. Solution of ligand. Here you can see this is the column. This is the column containing this is the resin. These are the resins, and you can see the black dot. Black dot. That is ligand. That is ligand. I have said that. Example, nickel. You assume that is the nickel. Okay. So you are passing all these proteins, and you can see that only the red protein specifically bind to your ligand. Red protein specifically bind to your ligand, and remaining all blue, all those proteins they have eluted out. You can see all these are which are not bound to your ligand, they are eluted out. And finally, by increasing the ligand or by increasing some ionic strength, you can collect your desired red protein. So this is the desired red protein. So this is the and we have gel filtration chromatography. Gel filtration chromatography. How do you perform gel filtration? So it's simply molecular sieving or gel filtration. So mana illallo we will be sieving voltage. Jallada pattern. So what is the principle in that? So the bigger molecules stay above, and the molecules that can pass through the pores, they will come down. They will come down. So in the similar manner, here the gel filtration column. So it is bound, it is filled with the resin material, or it may be agarose, or it may be polyacrylamide gel, whatever it may be. So they contain gel beads. Gel beads, and these gel beads are having small pores. Gel beads are having small pores. And whenever you pass the solution, so the porous polymer beads. So this is the gel bead, and they are having the pores. These are the pores, and here you can see this is a small red and big red. Small one and bigger one. You will be able to see those two are the proteins. Those two are the proteins, and this protein is entering into this gel bead. The chinna proteins they have entered into the gel beads, whereas this big they don't enter into the gel beads because they are bigger in size. They are bigger in size. So whenever you pour this, so here the small proteins they enter into the beads, whereas the bigger proteins they don't enter and they will bypass the beads. They will bypass the beads. That means if you take two beads, two big beads. So this is one bead, this is one bead, this is one bead. And I I am saying that these are having small pores. The beads are into small pores. So the small ones they will enter, whereas the bigger ones they will come here and they will try to move these things and they will come out like this. That means they don't enter into the beads and they will bypass, they will move in between the beads. So in gel-filtration chromatography, which molecules will come first and which molecules will come last? So that means bigger proteins will come at what time? First or last? Smaller proteins will come last. So the bigger proteins, because they don't enter into the beads, so they will move in between the beads so that they will come out first. Whereas the smaller things, they will come out last because they have to travel so much distance. So each and every bit they will enter and they will travel. So they will come very slowly. They will come very slowly. This is the principle of the gel-filtration chromatography, wherein mixture of proteins, whenever you have loaded onto this column, you will see bigger molecules they will elute first, whereas the smaller molecules they will elute very last. Very last. So it all depends upon the size of the protein. Size of the protein. And isoelectric focusing. Isoelectric focusing. In this isoelectric focusing, so in the earlier gel filtration you are seeing a column which is filled with gel beads. And here in this case, so in isoelectric focusing, the protein sample is electrophoresis through a gel having stable pH gradient. So what is a pH gradient? So what do you mean by pH gradient? pH gradient and area. So gradient is nothing but slowly increasing in the concentration. So here also we are saying pH gradient. So it is arranged in such a manner, pH 1, 2, 3, 4, 5, 6, 7, 8, like that up to 40. Up to 40. So that we call it as gradient. It is not having uniform pH. It is having different pH at different points, which we call it as a gradient. So the pH in the gel increases linearly from anode to cathode. From anode to cathode. What is the charge of anode and what is the charge of cathode? Cathode is negative, anode is positive. And we are saying pH in the gel increases linearly from anode to cathode. Cathode is negative. There you will be seeing more pH, which is positively charged. So each protein in the sample will migrate in the pH gradient and will get stacked at the position corresponding to its isoelectric point. So isoelectric point is a pH at which the net charge on the protein is 0. So when the net charge is 0, even if you apply the electrical field, it won't move. It won't move. That means depending upon its isoelectric point, they will stop. They will stop. And not all the proteins they will have the same isoelectric point. They will be having different isoelectric points, so that based on the isoelectric point we can separate them by means of this isoelectric focusing. And generally this pH gradient is prepared by mixing ampholites. Ampholites. So the better example commercially available are ampholine and pharmalite. Ampholine and pharmalites from the bigger companies, because they are not produced by the smaller companies because they are high cost. They are in high cost. So if you see these ampholites, what they will contain? Commercially ampholites have been synthesized by condensation of amino acids like glycine, glycine, and amines of selected pKas. Amines of selected dissociation constant values. Because obviously they need to have difference in the charge. Difference in the charge. So by means of these preparations, they will be producing these ampholites having different pHs. So you can see here. So after separation, you can see like this, because the proteins get separated. So this protein is having some separately. What is the pH of this isoelectric point? Isogenetic pH of this. ఏ pH కి దగ్గరలో ఉంటుంది అది? This one. So if it is having acidic pH, then it will move towards acidic pH అంటే negative charge ఉంటుంది. Then it will move towards positive. Very a simple thing చెప్పండి. So if it is moving towards the negative charge, means obviously it should be positive. And when it has come here, its net charge has become 0. That means its Pi value will be higher. Pi value will be higher. That means positive. That is maybe 10, 11, 12 like that. Ultrafiltration. It is also similar to that of sealing. Sealing, because in this ultrafiltration you will be having a membrane. You will be having a membrane through that membrane. So this is the Avogadro tube, and this you are seeing is a column. This is a column. Just like you are seeing in your plasmid isolation column, in that column you will be seeing some membrane, which is made of silica membrane. Like that, here also you will be seeing a membrane, and whenever you fill this, and this membrane allows only some, because of their pore size, they will allow some of the proteins to move through the pore, and the bigger proteins they will retain up. So we have filled our protein solution into this column, and then we have spun. Just spin this column, and then you will see some of the solution it has come out, whereas some of the solution it has remained on the top. That means the smaller ones they come out, and the bigger ones they have stayed, they have stopped there. And then you just by inverting that column, you can collect that protein fraction. That means here you will be having different types of membranes. Simply we call it as, based upon the size, you will be having your membranes. 10 kD cutoff, 20 kD cutoff, 30 kD cutoff, kilodaltons. Protein mass or protein molecular weight is expressed in kilodaltons. So 10 kD cutoff, which allows only 10 kD to pass through. 20 kD cutoff, which allows up to 20 kD proteins to pass out. And 30 kD, which allows up to 30 kD to pass down. That means here you will be getting two fractions. So one fraction is containing small amount of small size, and the upper fraction contains bigger size proteins. It is also a type of purification. But many people they employ these two to this because the membranes are very costly. When compared with above processes. And then the criteria of purity of enzymes. So what does it mean? Criteria of purity of enzymes. What it tells? So after purification of your enzyme, further you have to confirm whether your purified fraction contains a single protein. It contains a single protein because it may contain contaminating proteins, or it may contain some contaminating ions, or it may contain some contaminating biomolecules other than protein. So for that you have to confirm that your protein is pure. So further we have number of techniques for determining the purity of an enzyme, and purity is nothing but the single protein that you have to see in your things, and these are the following methods which we can apply to check the purity. So determination of non-protein contaminants, determination of non-protein contaminants, and determination of protein contaminants. Protein contaminants which can be done by specific activity change, and the chromatography methods. Similarly, you can use ion exchange or gel filtration or affinity chromatography, just like we have seen, and electrophoretic methods, non-denaturing or native gel, polyacrylamide gel electrophoresis, SDS page, and isoelectric focusing. So we have done all the purification process. After every purification step, you have to take some of the fraction and you have to check in the gel. You have to check in the gel by doing SDS page. Then you come to know whether single band is present or multiple bands are present. Single band is present or multiple bands are present. And SDS page, while doing SDS page, you will also have one advantage. Suppose you are dealing with multi-subunit enzymes. Multi-subunit enzymes. That means an enzyme containing more number of fragments. So we have studied about the quaternary structure of proteins. Tertiary structure is for only single polypeptide. Quaternary structure is for multi-protein, which contains more number of subunits. More number of subunits. And in the presence of SDS page, because these subunits obviously they might be interacted by non-covalent interactions, and moreover by means of what bonds? Disulfide bonds. So you know that insulin hormone is there. Insulin hormone is protein in nature. It is made up of two subunits, A subunit and B subunit. And these two subunits, they are interacted by disulfide bonds. They are interacted by disulfide bonds. When you run this insulin hormone in SDS page, how many bands you will get? You will get a single band or a double band. Because while doing SDS page, in the sample buffer you will add beta mercaptoethanol or dithiotritol. Beta mercaptoethanol or dithiotritol. So which brings about the reduction of the disulfide bond. Reduction of this 16 bond, so that that bond is clean, so that you will see separation of the bands, and that's why you will see two bands in SDS page if you run insulin hormone or insulin protein. So that is the fetching. If your protein is having, or if your enzyme is having multiple bands, then you will see more than one band, more than one band, more than one band. So that means here you have to purify by this ion exchange or affinity or gel electrophores. And when you run your denaturing, not denaturing native polyacrylamide gel electrophoresis, there you will see only one band. There you will see only one band because you are not denaturing the enzyme. And in this gel, when you separate your enzyme, even you can use the gel for checking your activity, for checking your enzyme activity, because in this condition it is not denatured, and it is functionally active. Whereas in SDS page it is denatured because you are using SDS. Obviously SDS denatures the protein and it will produce negative charges to the protein. All negative charges to the protein, so that it is moved towards the positive electron. And in this separation, the proteins they obviously separate depending upon their size, depending upon their size or molecular weight, depending upon their size or molecular weight. So in all these electrophoresis methods or these chromatographic methods, if you are getting a single band, then it gives that your enzyme purity is eligible. That means it contains pure single band. Pure single band. So this is one of the critical steps after purifying your enzyme. You have to check for the purity, and then only you can go for characterization. Then only you can go for characterization. So you will be having characterization of enzymes. How do you characterize? Suppose you assume that you have isolated a novel enzyme. You have isolated a novel enzyme. Then first of all you have to know what is the function of that. What is the function of that? Then only you could able to characterize further. And now you are doing your practicals. Isolation of alpha molecules or beta molecules. Isolation of alpha molecules or beta molecules. Suppose you assume that you have isolated and purified your enzyme. You have isolated and purified your enzyme. Then what you are going to do with that enzyme? After that, what you will do? So after that, whatever the things we are doing, they are the characterization. Characterization. So these are the characterization studies that we have to do. Native molecular weight determination and the subunit molecular weight determination indicating whether the enzyme protein is monomeric or oligomeric. And if it is oligomeric, whether it has identical subunits or different types of subunits. If it is oligomeric, there is one more problem. They may contain same identical subunits or they may not contain identical subunits. Because in the insulin hormone, you are having two subunits, they are not identical. They are not identical. That's why while doing SDS page, you will see two bands. Suppose you assume that if they are having identical subunits, how many bands you will see in SDS page? If they are having identical subunits, how many bands you will see in SDS page? Identical subunits I am saying. By using SDS and reducing agent, we can cleave the disulfide bonds. But we are saying they are the identical subunits. So if you run SDS page, you will see only one band. Only one band, not the two bands. So enzymes having identical subunits, they cannot be checked by SDS page. We have to apply other method. And number of sulfur hydrogen groups determination. Number of sulfur hydrogen groups determination. What is this sulfur hydrogen group? Sulfur hydrogen group and SH. SH group we call it as sulfur hydrogen group. Where it is present? Which amino? It is present in cysteine. Cysteine contains the sulfur hydrogen group SH. So in its reduced condition. If it is oxidized, that hydrogens are removed and two cysteines they will bind. Two cysteines they will bind. We call it as oxidation of the cysteine to cysteine. So that's why it is important to identify the number of the sulfur hydrogen groups present in the enzyme. And absorption spectrum indicating whether it is a simple protein or has a cofactor bound. Because many of the enzymes they have either coenzymes or cofactors. Coenzymes or cofactors. If they contain metals, then we call them as cofactors. If they contain biological molecules like NAD, FAD, then we call them as coenzymes. Coenzymes. If they are only metals, we call them as cofactors. And next, end group determination. End group determination or the end amino acid. Because if you take each and every enzyme or a protein, they will be having N-terminal and C-terminals. Having N-terminals or C-terminals. And during this end group analysis, you can You will also know that if it is having more number of subunits, then the end group will be varied. The end group will be varied to each and every subunit. So, native molecular weight determination. We have several methods, not the biochemical or the, we will be having some physical methods like light scattering method, osmotic pressure method, and depression in freezing point method. Generally, these are not used commonly. Earlier, people have used in the physics, and in our biochemistry, generally people will use gel filtration and ultra-centrifugation methods for determining the molecular weight. So, for subunit molecular weight, if at all they are having more number of subunits, they can also be determined by using SDS page. You know that while doing the SDS page, you will be loading molecular weight markers, and you are getting the proteins separated. So, you can compare with that molecular weight marker, and you can give the molecular weight of your enzyme. And we can also apply gel filtration to determine the molecular weight. How can we use gel filtration? Based on size, they get separated. During gel filtration, based on size, they will get separated. The bigger molecule they will separate first, and the lower molecule will separate last. And if you want to determine, then you have to run molecular markers in the gel filtration. So, known molecular weight markers, first we have to run in the gel filtration, and we have to collect their elution volumes, all those things, and then you have to run your desired sample or desired enzyme, and by plotting a graph like this, you can calculate. Just like మనం estimation చేస్తాం కదా, based on the standard curve, you will get your test sample. You will get your test sample concentration. In the similar manner, first of all, we will run the known molecular weight markers. After that, you will run your desired purified enzyme, and by plotting a graph like this, based on their BE or BO. That means BE is elution volume or void volume. So, based on that elution volume, that means at which fraction your enzyme is eluting, and at which fraction your standard proteins they are eluting, or known molecular weights are eluting. And if you plot the graph, then you come to know, and this fraction is having this molecular weight, and my fraction is also eluting at this particular fraction. So, its molecular weight will be similar to the standard one. And SDS page. So, obviously, molecular weight markers you come to know, and there also you can do a plot by movement, distance traveled by this protein, just like chromatograph, partition chromatograph, where you will calculate the RF value. So, known molecular weight, RF values, and your protein, purified protein, RF value, then you can plot the graph, and you will be able to cut your graph, and so that you will get your molecular weight. And determination of sulfur hydryl groups. Determination of sulfur hydryl groups, SH group. For that, we will be having some specific reagents. So, in the enzyme protein, they are estimated by using dithiobis nitrodenylic acid (DTNB) solution, by using DTNB titration. So, to the enzyme solution, generally we will add in the spectrophotometer. DTNB actually it absorbs at 412 nanometers. So, whenever it is interacting with the sulfhydryl groups, so you will get absorption spectrum. And if you are having more number of sulfhydryl groups, more amount of DTNB is bound, and you will get more amount of absorbance. And from there, you will calculate just like how do you calculate your standards with test samples. Similarly, you can calculate. So the number of sulfide groups will be calculated using molar extinction coefficient of that particular substrate. Molar extinction coefficient, and this will give the value of free or expressed sulfhydryl group. Free sulfhydryl groups. And if they are exposed out, they can be directly done like this. Suppose if you have any sulfhydryl group, they are present inside. That means indirectly you are measuring the number of cysteine residues present in the enzyme. If they are used in the cystic bonds, you cannot do that. If they are exposed outside, then only you can do by using this. If they are present inside, because if you take the three-dimensional structure of an enzyme, so inside the enzyme you will be having some cysteine residues. So in order to make them out, generally we will treat with SDS. We will treat with SDS so that the non-covalent interactions are removed. At a tertiary structure only, so that you will get a primary structure where you can see all the cysteine residues, they are exposed out. So then sulfhydryl groups we can further estimate by using the same DTMB solution. Absorption spectrum of enzyme. Generally absorption spectrum can be done in this range 200 nanometers to 950 nanometers. And generally you know that simple enzymes. So proteins they will absorb at 280 nanometers or 260 nanometers. Always doubt. So 280 nanometers. So nucleic acids will absorb at 260 nanometers. And suppose we will be having some different absorption than 280 nanometers. That means that along with the enzyme you are having something. It may be a coenzyme or a cofactor. So your enzyme is bound with coenzyme or cofactor, which can be determined by means of this absorption spectrum. And amino acid composition. Amino acid composition. You might have studied about the automated amino acid analyzer, wherein it works on the principle of ion exchange chromatography. So whatever the enzyme is there, you have to hydrolyze that and you have to fill into the automated amino acid analyzer. So there it will determine the amino acids that are present in your sample, and they will give the reading in the form of colors. For each amino acid they will give a particular color, and by that you come to know the amino acid composition. Because in the automated amino acid analyzer already they have the data for standard 20 amino acids. For the standard 20 amino acids. So by comparing with the amino acids, they come to know what type of amino acids are present in your protein sample, in your enzyme sample. So by that they will identify the individual amino acids. And L groups, because amino group and carboxyl group, so they can be determined. So amino terminal amino acid can be determined by using specific reagents, 1,4,2,4-dinitrobenzene and or phenyl isothiocyanate or dancine chloride. These are some of the reactions for N-terminal amino acid. And C-terminal amino acid can be carried out by hydrogen of lithium borohydrate or carboxypeptidase method. Carboxypeptidase is an enzyme which cleaves from the C-terminal. Whereas we have another enzyme called aminopeptidase which cleaves from the N-terminal. So by this you can identify the C-terminal amino acid or N-terminal amino acid. So this is a table. So after each and every step we have to check for the total protein, enzyme activity and its specific activity and the yield. We will do all these things. So here they have given a table example for isolation and purification of alpha glucosidase from beans, from plant source. So first of all they did extraction and they did saturation to 90% with ammonium sulfate. 90% ammonium sulfate said because the bigger protein might be required that much amount of ammonium sulfate. And then they did precipitation with polyethylene glycol. And this precipitate is then solubilized by the specific buffer. They have checked for the protein and activity, specific activity. And after that, by means of specific activity, you can see that specific activity always increases and the amount of the protein is going to be decreased at each and every step. So if amount is decreasing means the particular enzyme amount is increasing, the unwanted proteins are decreasing. At each step you are seeing here it is having more amount of this, and whenever you do saturation in this 90%, all the unwanted proteins, many of the proteins they can be removed. So you can see 50, not 50. So much amount of protein is reduced, and from here to here also you will see so much amount of reduced. And like that here you will be seeing only very little amount of protein. If you compare the amount of enzyme in this, so this portion contains more amount of your desired enzyme. That's why you will get more amount of specific activity. More amount of specific activity. And this is also a enzyme, phytate degraining enzyme from microbial source, from fungus, aspergillus, niger. Here also they have used from crude extract of ammonium sulfate fractionation and ion exchange chromatography, gel filtration and gel filtration. They have followed and they have checked the specific activity. They got maximum at this step. You can see protein is very much reduced, and this is the purification fold. So for each and every step, how much protein is purified. So by fold they will give, and the recovery finally they got how much. So this is the purification table. So remaining things we will do in the next class.